I am John Bradley. I'm the director of the Gut Reaction Data Research Hub. And today we want to take you through our pathway to sustainability. If I could have the next slide. And the next slide after that. And then the next slide. The Gut Reaction Data Research Hub is about collating data on patients with inflammatory bowel disease and making it available for research. The overall goal is to collate data from NHS trusts, and you can see the hospitals that we're working with at the top of the screen, and also from NHS Digital, the Inflammatory Bowel Disease Registry, from academic collaborators that have research data, and pull all that data together into a secure cloud environment where it can be made available for research. And we're going to be taking you through that process and how we will embed it for the long term during the session. Thank you to everybody for joining. Very aware that the audience is wide-ranging, and I would ask speakers to bear that in mind during their presentation. We do have sessions for questions and answers, and you should be able to see on your screen the ability to put questions either in the chat or the Q&A section, so that we can then address them either through the chat or by directing them to the speakers during the Q&A sessions. I'm going to now hand over to Mark Avery, who is the Operational Director for Gut Reaction, to give us an overview of our approach to sustainability. Mark.
Thank you very much, John. Hopefully you can see me. Very good. So firstly, thanks from me also for joining. It's great to see so many people on the call. So thank you very much for giving up your time for us this afternoon. So I'm going to give a quick overview now as a sort of an introduction to this sustainability session, and that is the core focus of our presentations this afternoon. If I start by just outlining the three themes or the three main aims of the Gut Reaction program. So next slide, please. Okay. So aim one is essentially to be building on the data that's already held by the NIHR IBD bioresource, and critically, this isn't just the data, or critically, it's the consent given by individual participants enrolled in the bioresource that have given their permission for their health and care records to be accessed for research purposes. So core data is already held by the bioresource, and the programme is specifically looking at supplementing that with data from additional sources, and these include NHS Digital, HES data for those that are familiar with it, but hospital commissioning data, essentially data from the IBD registry, and particularly longitudinal or data captured over time from electronic patient records at NHS trusts. So as John outlined on the previous slide, we're working with 10 trusts in the first cohort. Discussions are now underway with a further 13. And this involves working with them and the governance in individual organisations to get really quite rich health and care record information, discharge summaries, outpatient letters, pathology lab results and prescribing information, which offer huge potential in terms of understanding disease progression over time and interaction with treatments. Next slide, please. The second aim is to make these integrated data sets then available via a secure cloud trustworthy research environment, or TRE. Many will be familiar with that term, which essentially involves bringing the NHS data together within the NHS firewall or the HSCN network, and this is identifiable data at this stage. That means it can be linked, and then subsets of those data to answer specific agreed research questions for access by specific agreed organisations can then be de-identified and made available in the TRE. And there are a couple of options there delivered through our partner AIMS, or scaled up in an Azure environment. Sorry for the dog barking in the background. Next slide, please. Okay, so in terms of just outlining the discussion for this afternoon, then the only way that that this approach works, and the only way the hub can be sustainable, is to make sure that we are meeting the core needs of all of the stakeholder groups that are required in order to be able to make this work. So we've got the overarching aim there to ensure that the approach that we're developing is acceptable, scalable, replicable, and financially sustainable. And really key to unlocking all of that is by making sure that patients themselves are integrated and involved in decisions about how their data are used, and that they're comfortable with what we're doing and how we're doing it, and who is accessing the data for what. So the central convening theme here is to make sure that everything that we're doing has an overriding patient benefit. The 4 Ps there are provided by the sort of P4 Institute in Seattle, so they're not my invention, but they're really good in terms of highlighting what the ultimate benefits that we're seeking to achieve here in terms of improving prevention, prediction, increasing patient participation, and development of personalised treatments for patients. So we'll be hearing shortly from Tudor and Sarah about the patient perspectives in all of this, and it's absolutely critical to get that right and to make sure that this is central in order to be able to unlock everything else. Obviously, there needs to be intention there for a wider population or societal benefit from what we're doing, and everything that we're doing in the hub program is funded ultimately by taxpayers through the Industrial Strategy Challenge Fund and UKRI. But making sure that that's a good investment and showing the return on investment from a public and taxpayer point of view is important, and not always the same as the patient view. So we need to bear that in mind. It clearly needs to work for NHS organisations in order for them to be able to be prepared to share data and to be able to do so within the strict governance requirements that each are operating within. And ultimately, in terms of then use of the data for improving our knowledge of disease and developing new products and services, the manner in which we make those data available, whilst addressing the sort of the interests and sort of constraints, if you like, that need to be in place in order to be able to protect the interests of other organisations. So really, that's what's behind the trustworthy research environment, or TRE type approach. And underpinning all of this, I'm not going to go through them in detail now, but many of you will be familiar with the 5 safes model for ensuring that. Okay, so in the interest of keeping to time, I'll hand over shortly to Sarah and Tudor for the first session, but we will then be hearing from Neil Walker as the Chief Data Officer and Alex as a researcher in terms of the practical experience of pulling data together and analysing it within the TRE, and then we will pass to GSK colleagues at the end of the session for the industry perspective on sustainability also. Okay, so thank you very much. That's all from me for now. And Sarah and Tudor, if you could come on, I will hand over to you now. Just a quick reminder for everybody, if you've got any questions as we go, please post those in the chat. Thank you. Sarah.
Thank you, Mark, and thank you for this opportunity to talk about how the patient and public perspective is really at the heart of this programme. And I think for those of you who have noticed the recent debate around the GP new database, you will know how important it is to get the patient and public trustworthiness of a big data project absolutely right. And I'm really delighted that it's a key feature of this programme that we do get that right. So what we're going to use this section for is to really have a kind of Q&A between myself as the executive lead on the PPIE element of the programme, and Tudor, who has been on the journey with us since the very beginning. Tudor is a member of our patient advisory committee, and he has a particular interest in the financial sustainability model of the programme and how we interact with our various beneficiaries and partners in that. So I'm not quite sure how this is going to work and whether Tudor's going to pop up alongside me somehow. So Tudor, can I just very briefly get you to become visible? There we go. And just to very quickly intro yourself, and then we'll get going with the Q&A.
Yes, thank you, Sarah. So my name is Tudor. I am one of the patient representatives. I was diagnosed with Crohn's about 2 years ago, and that's when I started looking into the condition, and I found out about the charity, and I found out about this research program. So that is my background.
Great, thanks, Tudor. And you know, one of the critical elements about the approach of involving patients in this program is about building trust. How fundamental do you think that is to developing a successful outcome with the program and in big data generally?
Yes, trust and transparency are very important, because health data is invaluable, especially to us, the patients who generate it. So whenever we hear news of our data is going through this change or is going to that place, it's a bit scary. So that's why it was very good in this program that patients were approached from the beginning, and we had a very big patient group, and we saw not just we didn't just meet the people that are making this thing happening, but we also met some of the researchers and saw some examples of how the data will be used and what sort of processes are put in place for people to access that data. So that was very helpful, and it created a lot of trust from the patient side.
Great. And the patient advisory committee, and you're a member of that group, is obviously heavily involved in the program and looking at all the different aspects. Where do you think that committee has been able to add, you know, new and added value to the program, which might not have happened if the patients hadn't been in there? Right.
So I feel the group added value in two ways. First of all, how the public involvement is being run, and how we communicate with each other, how we contribute to the meetings. But second, it contributed specifically to the gut reaction hub, in that, for example, it defined red lines over how patients don't want their data to be used. We don't want it to be used for insurance purposes. We don't want it to be used for advertising purposes. We implemented some changes on the steering committees, which check who gets access to the data and how they get access. So in the way we did it, patients have a say with every access request. So nothing happens in the background, and I feel that is the way things should be run in general with data hubs.
I mean, that's interesting. Do you think that some of the lessons that we've sort of shared in the program could be learned from you know, the current controversy around the GP database? What lessons do you think they could learn from the approach taken in the program?
Oh, right. Well, that is a hot topic. The timing is very important. So if people find out about this from the TV as if someone is going behind their back, it just looks very bad, and it's much better to be approached first by the GP maybe, or by fellow members of the public to be informed. I think that that's a very important lesson. Also, have a clear understanding of where to go if you don't like, if you want to say something that doesn't agree with you, you need to be very clear where to go to put your complaints forward.
Yeah. So in terms of the next steps and the focus for the Patient Advisory Committee in particular, looking forward for the rest of the programme as we go into a period of looking at sustainability and so on, what's the focus for the committee in the next 18 months?
Right. So yes, we did reach a midpoint in this project. As patients, we were very happy to see the data already being used and bringing results. So now we're helping to get this this project to become sustainable in a way that agrees with all the stakeholders, because we do want to encourage research. We do want to encourage research that wouldn't have been possible without it. But we also want to maintain a high level of trust and about how the data is being used. So we want to make sure that the money that comes in respects our views pretty much. And it's an iterative process. We're improving with every meeting, so it's good.
Just finally, a quick one. You're actually a researcher by background, aren't you, on health technology products? What would you take away from this experience to your professional life?
Right. So yes, my at the moment in my work, we're focused more on hardware than on data. But yeah, I realized by being part of this how important it is to involve patients early on, and this is something we started doing. We started having discovery sessions with patients and other peers involved in their care, even before we start the project, even before we put in a grant application. So I mean, some things that patients may find it's trivial, just voicing it out in the right forum can have a big impact. So we started having these sessions early on, and budgeting and planning for the research project was also influenced, because I know this should be managed very carefully. Communication should be maintained throughout the project with patients, not just in one-off meetings. So yeah, this was very useful in that sense.
So that's some really good advice, I guess, for any company that wants to access the data in gut reaction going forward, that they, that whole ethos of involving patients from the very, very beginning, framing questions right the way through to getting the research answers is really important. Thank you very much, Tudor, for that. And I will pass over now to the next speaker. Thank you.
So we do actually have a little bit of time for questions and answers at this point. And we have got one or two questions that have come up in the chat, and a comment really embracing the involvement of patients and also welcoming the sort of wide range of healthcare professionals that are involved in the program. One of the first questions, and we may cover this a little bit more during the session: what patient data is in gut reaction? Have they given their consent? So the patient data I can address is quite wide ranging. It includes self-reported health and lifestyle data from the patients. It includes data that we can access through their health records. And then it also includes quite a lot of genetic data. Have they given their consent? Yes, they have. Maybe interesting to get Sarah and Tudor's perspective, because we don't just rely on the fact that they've given their consent. I think it's important that they're kept aware as the program progresses as to exactly what is happening with their data. I don't know whether you have any further comments on that particular question, Sarah or Tudor.
I definitely, from my perspective, working with a charity, communication around a constant communication, and actually this is a constant dialogue as well, is really, really important, if nothing else, because patient and the public views around the use of data has actually evolved fairly quickly in the last 2 or 3 years. And so you can't assume necessarily that what was once unacceptable is not now acceptable, and vice versa. So that communication and dialogue around consent is very key.
Thank you. Next question, how can people with IBD take part? Can I join? Well, the recruitment is through hospitals. We've got over 100 hospitals who are signed up to recruitment of people into the IBD bioresource that underpins gut reaction. So almost certainly you can, and best to discuss that with the physician looking after your care. Next one, what criteria for wider access? One thing to trust researchers you've met, but you may not know much about future researchers. Some research is collaborative between industry and scientific community. Are all your patients comfortable with industry having access, and are there specific constraints for them? I think here it's important, and whether Sarah or Tudor want to just outline it briefly, the mechanism we have for reviewing all of the studies that come to us, and the importance of involving patients and the patient advisory committee in that process.
Yes, I can answer this. So I was giving the example of some researchers we met that we get to see and understand the data, but every single access goes through 2 committees, and patient representatives are there. So every data access request has to say why they want the data, what they intend to do with the data, and how much data basically they want. And based on that, it goes through a formal process of being reviewed, and we can ask for changes. Yeah, we did case studies in which we reviewed and we're not comfortable with all the requests. So that thing will be reflected in the day-to-day operations of this.
Thank you. And I should just say that Neil Walker, who'll be coming up next, has just put a link in the chat to a site that tells you whether your hospital is part of the bioresource and gut reaction. So if you wanted to check, you can do that. There's a final question we're not going to do justice to, but what do you think is the right mechanism on disseminating results of data analysis directly back to patients or through other media? As I say, not do justice to, because we need to move on to the next session. But I think it is important, and we need to probably do more in terms of feeding back to patients the results that come through. Ultimately, in parallel, we need to publish results in peer-reviewed journals so that they can be used by academia and industry ultimately to change practice. Do keep putting questions in the chat or the Q&A session. I'm going to hand it back now to Mark, who's going to introduce the next 2 sessions.
Thank you very much, John, and Sarah and Tudor for that first session. And obviously the questions coming through. So do keep posting questions in the chat if you have them. We've got a dedicated slot after the end of each speaker to try and address all we can in the time that we've got available. Okay, so pleasure now to be able to introduce both Neil and Alex. So I think if we can move the slides on a little bit, that's it. So so we're going to hear from Neil and Alex. Neil is the Chief Data Officer for the Gut Reaction Hub, and Alex is a postdoc fellow working at the Sanger Institute. So it'll be great to hear about how this works in practice now. Neil's going to take us through what it means in practice in terms of pulling the data data together to make it available within the TRE environment, and then some perspectives from Alex on a really sort of fairly complex COVID study that involved multiple data sets, and his reflections on the initial findings and what it was like to use the gut reaction research environment. So thank you very much both. Neil, if we could come to you first.
Yes, so before we move to the next slide, just to say, to following John's previous point, one thing we are trying to do at the moment is commission a participant portal so we can feed back results more directly to our participants and also advertise studies they may be interested in joining positively. I suspect we'll end up with a model where participants will actually pre-select whether they engage with a particular thing rather than finding out that it's happened or relying on the pack, but that's something that will happen in the future, and this is a very very changeable field at the moment. Anyway, we have the next slide. I'll talk about this stuff. So we advertise the data sets that we have through the HDR UK Innovation Gateway, which is a relatively new thing, and this is a relatively old slide, so no doubt it's out of date. But the idea is that you can search for any data sets you like and see who is talking about the things of interest to you through the search function. We are represented as one of the collections there. We have 11 data sets, and it's a growing resource, and gradually they're building in requesting mechanisms that really work to reach out to the people behind this data. So if you could have the next slide, please. So this is what it looks like when you get in. At the very top level, you have a list of items. These are, so a case report form is basically what a clinician might recall, or the clinical care team. Health and lifestyle questionnaire is what a person might say about themselves. And you can see these are named as data sets. And if we move to the next slide, please, you can see that you can drill down, and on that, you can look at technical details. You can look at whether the data is any good, the data utility, is it changing very much? Is it cleaned up? Is it well described? And also you can say, find out other things about it. You can also get quite a lot of detail there. But that drills down on one. So if you move to the next slide, please. This is lots of detail, but so far you can only do one data set at a time. So if you are interested in, have I got data on from the clinical data like this and the person's life story like that, it can't be currently achieved through the gateway. I dare say the gateway will do it because they're moving incredibly quickly, but at the moment you can't. So if we look to the next slide, this is where it gets a bit technical. So what we have is a data warehouse where we put the data all coded up together. So you've got not just one data set, but several data sets just described in this data warehouse. And this is using a thing called I2B2, which is quite a popular method of looking at healthcare data. It's based around the concepts that are familiar from the NHS. So the SNOMED CT is what the NHS uses to give a code to everything that happens in a hospital. And so we've coded things back into that from the questions so that people can combine several data sets together to do a query across the data sets. And if you go to the next slide, you'll see this is what happens. So you can imagine picking bits out of the different pieces, and it tells you whether in the data set or not there are any patients who have that requirement. So in particular, this could be the difference between whether you do or don't do a study, because it's one thing to see you collected 200 questions about a person, but you don't know the answers. But if in here, you can say, well, I can see that enough people answered that question positively that I will do my study with this group of people. I will drill down and know I have enough people there to be worth doing something. And so the discussion I had with the Patient Advisory Committee last night was, how do we make this available to researchers? And their view, broadly speaking, was, if we make this not too disclosive, as in we hide some of the more secret or not secret, the rarer events, that could be more public. If it's going to be this rare, it has to be pretty secret, because you're going to end up, if you're not careful, with very small patient counts in here because of the rare things that are recorded against a different patient. And there is some sensitivity. Yeah, obviously we don't keep the personal details here, but if you have enough diagnoses and medications and so forth, it looks pretty individual after a while. So the idea is there may be one view that's extremely limited but publicly available, and then all the detail is hidden underneath, and it would go through the data access application route that Tudor described earlier, where the person is reviewed, who is applying, what are they doing it for, what do they need to see, what do they intend to do with it afterwards, all those good things. So if we go to the next slide, and if I could have some questions about that, if there are any.
Thank you, Neil. So there is one. So somebody's asked, can anybody look at the data sets, or is it just researchers?
Okay, so I think what we'll do is we'll end up with a top level bit which give the broad outlines. So for instance, sort of age bands, sex, broad disease diagnoses, a few things like that. So you have a rough idea what the cohort of patients look like. And that could be public, because you're only going to get answers in the sort of hundreds or whatever. It's going to be big numbers of people. It's just a count. It doesn't tell you who anyone is. It doesn't tell you where they live. It doesn't tell you any details that are confidential to their medical history. It's just very broad characteristics, just to show what the cohort looks like. But then when it comes down to the detailed stuff, it's going to need at least a registration, so we know exactly who's doing what with it. And I think the direction of travel is it will actually have a full data access application and live inside this trustworthy research environment, in order that if people find something, it's not leaking out at all. That's the difference between these trustworthy research environments and other things, in that downloads are not permitted at all. So even though you've signed a contract saying you will not try and identify participants, even though it is you're required not to try and drill down that deeply, it also is not permitted. So it's a method of enforcing constraints on you, technical constraints.
Thank you.
Yeah, so the answer is yes to both.
Yeah, and there are the different layers, aren't there? So the I mean, as you outlined, but the core sort of data set descriptions are available via the HDI UK National Innovation Gateway, which at least describes the data set and what's in it. But yeah, certainly when it comes to then looking at the cohorts within it,
Yes.
I mean, it provides a brilliant interface to allow researchers to understand what's available without getting at that underlying information. So with that in mind, and you were talking about, as you get more and more into the detail, sort of the data becomes sort of potentially more disclosive. So for cohort discovery use, what level of aggregation do you think is sort of workable to try and, or what steps can be taken as this develops to try and protect the participants, or the ability to be able to identify individual participants from the data?
Well, there's two aspects. So one of the aspects about keeping the data under our control is we know that other people aren't trying to link other things in. Because lots of times where you've seen a report where someone's been identified from the data that's been released, it's because someone knew something else. So famously, when the NHS Digital have a thing called hospital episode statistics, and when they first released that, they thought this is anonymous enough, but they didn't realize that because everyone goes through the NHS, that includes famous people. And so people know other things about famous people. So if a famous person turns up in hospital, it's news. So you knew when someone went into the hospital, so you could actually pick them out of the data by that. So that was a terrible wake-up call, that merely because most of us are fairly anonymous in the public, you cannot just release this data because people know other things and will do other things with it. So so that's the first bit. I think there is an idea of utility in data that says you can squeeze the identifiability out of it, so it gets more and more and more into just counts of big numbers of people. But at that point, you start losing all the detail, which is what you actually want. So for instance, many of our researchers will be interested in individual drugs, and the drugs are going to be given in weird combinations because there is a sort of method. You do that one, then that one, then that one, and that's going to become unique quite quickly. So I think to accept the fact you cannot de-identify completely, but make sure you really do know who the researcher is looking at the data. You don't know what they said they're going to do with it. You make sure they can't take it away. Those sorts of things are the protections. And that's what the 5 safe bits is that we were talking about, which is to make sure you can rely on that there will be a public benefit from this research, and you also have technical controls to stop people downloading it, if that answers the drift of the question.
I think so. No, that's great. Thank you. Okay, so another one from the chat. How can researchers determine whether a data set might include the rarer condition which they might be interested in?
So I think if they're looking for the other model we have considered is like a service model. You just ask us, and we run this tool for you. So there is an idea that people would apply to come in because they want to do some exploratory stuff. Because I've never met a clinician yet, if you show them that, who won't ask another question. So I got down to 735, and they'll go, oh, if I added a third thing, would it get down to 500 or whatever? So I think there is an element of lots of people want to try it themselves, but there is also, we could run this as a service model and just say, ask us your questions. We will then use this thing ourselves and just tell you the answer. It's particularly the case because there are hundreds of fields in these data sets, hundreds and hundreds, and some of them will be fairly sparsely populated. So therefore, that is a sensible thing to do, to say, do you have people like this? And we get a lot of inquiries like that now. And we just go and rummage through using the tool ourselves.
Thank you. And what assurances do you need in terms of people requesting access to the tool? So if we're thinking about safe people, who are outlining in the sort of the 5 safes a little bit earlier, how is that sort of determined in terms of who can gain access?
Well, so hitherto there is a concept of a bona fide researcher of sufficient seniority who is working in the field. So that's a sort of combination of things. So there is a move afoot to say you should have had a particular training, and that may be the thing that comes about in the next year. So the Office of National Statistics offer an approved researcher scheme in the UK for people who've taken the relevant training. We, on the other hand, are more open to people worldwide, perhaps, to come in and ask the questions. We know, for instance, many of the pharmaceutical companies have bases in America, even if they're analyzing the data here. So we have to be a bit more open than that. So we look for someone, at least the primary applicant, they may have a student under them, but the primary applicant to be working in the field, to have publications and publication records that show they know what they're doing, to approach us via their institutional address and not via a Google or some other type of thing, so we know who they really are, who they say they are. And when people apply, we also have institutional sign-off from someone in their institution who is legally able to sign that they can sign that contract. So that means that their institution is taking responsibility for their conduct as well. So that is the framework around it. But I suspect what will happen gradually is there'll also be a requirement to register as an approved researcher with somebody. But I've made this example before that others may have heard. I was on a data access committee where we had an absolutely top chemist come to us to ask for genetic data, saying, I'm really top chemist, these are all my publications. Genetics look quite easy to me, so I'll have a go. And we said, no, please go and find a geneticist to go and work with in order that they can explain what it is you're doing. So the idea the person could be approved, but not to do this thing. So it's not the idea that a person is inherently approved. They have to still do the right thing and still come through with the credentials in the institution and so forth.
And we're pretty much at time, so there's a couple of other questions in the chat. I'm keen to make sure that we give time to all of the speakers. So the two outstanding questions that we've got are around whether there's a free model available for running a search as a first step before applying the data. And to answer that one quickly, so yes, absolutely, you can sort of get in touch and test that viability before making a formal application, and that's really what this tool is designed to deliver. And another one about just what are the risks for patients about data being used in this manner. And again, this is designed as an intermediary step, if you like, to be able to reduce that risk. But depending on the type of question being answered and the level of granularity required to answer it will depend on that depth. But we'll,
yes.
Yeah, I don't know, Neil, whether, so in the interest of time, whether you might sort of consider just answering those in the chat afterwards. We'll
yes, I'll just say that a data access committee must be able to say no. So if someone comes through with a purpose which you don't agree with, you have to be able to say no.
Noted. No, absolutely, a really important point. Okay, so if we could move on to the next one, and we're still going to be have Neil sort of presenting for this next section, and I was getting ahead of myself in terms of introducing this as in the previous section. So if we could look now at the COVID-19 case study, and Neil will talk first about bringing the data together, and then we'll move to Alex from the Sanger Institute talking about what it was like to use the environment and the findings from that study. So Neil, back to you.
Yes. Okay. So if we could have the next slide, please. This is my. Okay. So the one after that then. Yes. So one of the things that happened during the pandemic is there was suddenly a real energy to accelerate access to linked data. And in particular, anything to do with COVID could be achieved and was achieved. So HDR UK were very, very good to go out to their hubs, other contacts to say, look, we're talking to these public bodies. Shall we help you get access to data in order to enrich what you have already, in order to do important public health work? And so the first people that we got on board with Public Health England, and they're the ones who had all the COVID-19 testing data. In the beginning, there wasn't very much of it, but they got onto it very, very quickly. And so we signed a contract with them, a data sharing agreement to say what we were allowed to do with data, what other people were allowed to do with data, all those things we've mentioned. So that was an very early win. And then that we also started getting in touch with the Intensive Care National Audit and Research Centre, which is ICNARC, and they send information about who's been in hospital, what's happened to them in hospital and so forth. And that was in receipt from 1st of December, so quite a long gap. But we got there and it's in there now, and Alex will talk about that later. The third thing we did is we got access to what's called the NHS digital spine. People may have heard this who have an NHS background, but it's the bit that allows any hospital, any doctor to look up somebody if they stroll through the door, or even worse, if they come through the door unconscious. It's their personal demographics lookup service. And we've been able to use that, again, since early April to make sure we find out if someone's died or if someone's moved or we can't contact them and so forth. So that was also very, very helpful. And we also got hold of the data, sort of central administrative records about hospital admissions, outpatient visits, critical care, A&E, which is collectively called Health Episode Statistics, or HES. And we got that early this year. That was a bit of a beast as a contract, but we got there. And that's one of the reasons, again, is just because it was a pandemic, they did not say, oh, you can have the data. They put us through all the same processes as you would go through normally, including their very, very influential patient safeguard committee called IGARD, which I suspect is why NHS Digital did not notice the problem with the GP data that's just happened, because they knew that their committee would be looking after the data and they feel comfortable with that, but they haven't explained to the public, which is why there's a problem. Okay, next slide, please. So what we did is we started putting data into our trustworthy research environment. So what the blue box in the middle is meant to represent is our data center, where data is still identifiable and data flows into it. But what we do, which is perhaps different to others, is we don't allow the researchers into that at all. not even to a bit of it. They actually have a separate thing, which is not part of our data center. So what we're doing is we curate it, we make sure everything joins up properly. We use the Privitar privacy enhancing technology to do some de-identification work. AIMS is the data center, so there's a plug for them. They do a lot of stuff with the NHS. But the researchers come through and see a separate area which is really locked down very tightly to make sure nothing leaks out, and they don't get to see the original stuff either. If we have the next slide, I think we're over to Alex now. Yeah, thanks.
Thank you, Neil. My name is Alex Sazonov. I'm a postdoc at Sanger. I mostly work on various genomics analysis, but kind of in the earlier stages of the pandemic, I got involved in this project to actually look at the risks that IBD patients may face from COVID-19. Can I have the next slide, please? So this was really started in the very early months of last year, around maybe April, May, when there was a lot of anxiety about the potential effects that COVID might have on people with IBD who are sort of known to be on at a slightly higher risk of non-COVID infections, but perhaps that's still a research question that this might be actually mediated by specific therapies. But at the same time, there was a lot of hope around proposals to actually that some of the same immunomodulating therapies might be trialed as treatments against critical COVID. So we really embarked onto a project to try to monitor in almost kind of weekly basis whether we see any potential indications that certain therapies might be associated with higher COVID risk or lower COVID risk. And really, it's quite hard to study this using any data set, but actually, I think the bioresource cohort with its 31,000 patients was in a pretty unique position to do so. So as Neil said, this was linked to initially testing records from Public Health England and eventually with various other sources that included things like hospitalizations and death rates. So we started running some analysis, if I can have the next slide. So in very graphical format, the thing we tried to do is to, for example, we had an outcome of interest. So in this case is whether the patient ever tested positive for COVID. We adjusted this for a variety of relevant covariates that we sort of knew from literature and thought were sensible. And then we tested each individual drug one by one, whether the patient is currently prescribed it. And then we tried to calculate some test statistics that basically indicate whether there is a significant association between an individual parameter, which is the drug, and the outcome. So initially, we really tried to look at only the infections with the hope that in the future we will extend this to perhaps critical COVID, so hospitalizations, ICU visits, and deaths. But really, what we observed in the data that there weren't enough hospitalizations and death events to do formal statistical analysis on this. So we kind of limited ourselves to only being tested positive for COVID. And the things we found, if I can have the next slide, please, is that actually COVID amongst IBD patients looks quite similar to the average kind of national levels of COVID. So here you can see that across 31,000 patients, I think roughly a third of them had at least one test done, and around 1,600 of these had a positive test. That means that the positive test rate is around 5%, which is quite similar to the 6% UK average. I would say that this is perhaps not comparing apples to apples in a direct way, because we know that some IBD patients were shielding, others were suggested to exercise kind of enhanced social distancing. And of course, the data set itself is not ideal for this type of research, but I do think it's a very strong indicator that there's no massive effect of COVID on the risk of getting infected with COVID amongst IBD patients. And also, if you look at the plot above, here we kind of have a week-by-week breakdown of the number of, well, fraction of registered positive tests between the bioresource population and the Office for National Statistics data, which collects sort of countrywide data. And actually the distribution between the two matches almost perfectly, indicating that there hasn't been kind of any major differences in the shift, which I think if there were, might have been indicators that actually the IBD patients might have had slightly more severe response. Out of the sort of therapy analysis that we did, fortunately not much has come up as positive. So there is the only nominally significant association, statistically significant association, is that mesalazine appears to be very marginally associated with higher risk of infection, but I would really try to emphasize that the effect is quite small, and at this effect size and with this nature of the data, it's very hard to claim the causality of this association. Perhaps this is just some indication of biases in the data itself and not true risk effect. And to add, for many of the drugs, we really had sort of hundreds and thousands of patients on them, especially various biologics. And despite that, we really didn't see much effect, which I think is a positive indication that IBD drugs do not appear to be very strongly associated with any negative COVID outcomes. And then finally, unfortunately, of course, some of the patients who had COVID have passed away. In our data set, that's around 29 patients, or around 1.8%. Given the age distribution in the bioresource, I would say this is not very different from the kind of average national levels of death, which is tragic, but perhaps does indicate that IBD is not associated with much stronger or any different outcomes in terms of severe COVID. We're currently trying to validate some of these results by manually contacting 10 of the recruitment sites and asking them to perhaps provide any additional information they might have on their patients, and see whether that kind of their conclusions regarding who was hospitalized and who has unfortunately passed away match the information that we see in our data. Next slide, please. Sorry, I should probably add that the previous slide contains information from our active research project, and it can change. It hasn't been peer reviewed, but it was presented to various colleagues throughout kind of the analysis process. Finally, some of the experience that we had using the actual trusted research environment that Neil was talking about for analyzing data. So on the analysis team, I would say there were 4 core people, a combination of very talented and kind of strongly data-driven clinicians, epidemiologists, and me, somewhat geneticist. And we did an analysis that really is quite reflective of a lot of clinical trial analysis data, epidemiological analysis trial data. So we were able to kind of take up a lot of data sets that were usually effectively kind of tabular data. That's, think of Excel or just CSV spreadsheets. Then we had a number of different programs, scripts that we wrote that kind of ingested the data and quickly did the analysis, and with some code to actually produce these plots in an almost continuous manner, so that every time we got, say, a new public health England release with testing, we were able to rerun it and see if anything sort of suspicious pops up. As my personal opinion, I think I was really quite pleasantly surprised that despite this being a very locked down environment by design because of the important kind of clinical implications, it was really perfectly usable for doing this data science-y analysis stuff that we're used to. The team was incredibly helpful with helping us to install maybe computational packages and statistical things, and really facilitated the analysis very well. And I really look forward to seeing kind of the next step of this development when the platform becomes also usable for genetics and genomics analysis. With that, I think that's all from me. Thank you.
Thank you very much, Alex and Neil. So if I can start sort of feeding in questions. So it's one from a researcher who's currently working with data in a similar environment, so in a trustworthy research environment provided through the British Heart Foundation, saying, I'm keen to do some work on gut-related phenotypes. Are there plans to enable linking with other NHS digital TREs to make this possible? So this is a reasonably hot topic around trustworthy research environment sort of federation or linking these things. Neil, I wonder if we might come to you on that one. Would that be okay?
Yes, I would agree. It's a current hot topic. HDR UK are leading very strongly on this because groups do not want to let their data leave to go elsewhere. And if you don't want to do that, somehow you must join up between them by some other method. So the two methods of doing this that are currently suggested is effectively you write an analysis package that could be run in two different places, and then you bring the results together. Or the other method is that you actually permit people to see into two different of these TREs at the same time. So those are the two different models being proposed currently. I suspect we know that the former has been in use in social science for a bit. There was a group led by Paul Burton who were doing this in Newcastle. So I think that may mature first. But I think there are other things to be done with this TRE work. Like, for instance, Alex is asking about, can we get genetic data in? Well, we can, but maybe that's the case where it needs to connect to it held elsewhere, because moving around big slabs of data in like images and genetics is not sensible, it's not cost effective, it's not secure either. So it may be that we end up developing so we can do this federated approach. And I think the more people like Alex Handy who say they want it, the more likely it is to happen. Yeah.
Wonderful, thank you. Another question. So, Neil, what does OPEI mean?
I don't recall it. Let me see if I can find it. I will quickly have a look to see if I just accidentally covered something by mistake, which is quite likely. So yeah, that's yeah, I will come back to that. Yeah.
Okay, no, that's fine.
I did answer a question about the costs in the sort of chat bit, but I think one of the interesting things about these trustworthy research environments, if we move to a completely cloud-based system, it would allow you to charge people independently to use it. My view is that anything to do with that cohort discovery should not be a chargeable thing, because you want to invite people to find out whether they've got a study to be done. If they have, then that's when you might consider charging. And we certainly heard from one of the other hubs quite an interesting model yesterday, which is NHS goes free, academic is cost recovery, as in it costs us stuff to do, money to do things, and industry, you make enough money from them in order to pay back the thing you did with the NHS to make sure everything just keeps running. Just because the goal is to have a non-profit entity that actually is self-sustaining in the future.
And one more question just before we move on. So are there plans to provide analysis support for researchers who are not experts in big data analysis? And there's a specific particular question here a bit. Are there plans to offer that analysis at cost? But there are two separate questions there. So one is about the provision of analysis support, and the other one is what the cost model might be to go with it.
Okay, so I do know that one of the HDR UK Alliance members, the ALSPAC group, have a data buddy scheme, particularly for this, because they basically say, look, we are the ones who know the data. We're going to have to help you get the thing you want. out of all the data we've got, you want a particular thing. So they have a data buddy scheme, and that's one of their cost recovery models. So effectively, you end up paying for half a day of someone's time or a day's time or actually proper support. At the moment, though, if truth be told, we don't. We rely on smart people like Alex coming in and doing his own thing. So, yeah.
Wonderful. Thank you very much. So keeping to the times we have on our agenda, if we could, and thank you, Neil and Alex, both for those, and there's some very complimentary mentions in the chat as well. So could we move across now to hear from our colleagues at GSK? So we'll be hearing this afternoon from Victoria Higgins and Meg Elm. So if I could hand to you now, and really important and interesting to cover the industry perspective on this in terms of accessing data in the way that we've described, and still working in the public interest. So across to you, Victoria, I think, to start.
Okay, I'm going to go ahead and I'm going to go ahead and just go ahead and speak, since Victoria is having trouble speaking. So thanks for inviting us to speak today. I'm Meg M. I'm a geneticist at GlaxoSmithKline. And today we're going to talk about how important this data is to the discovery of development in therapeutics. So first of all, really at GSK, patients are the most important aspect of everything that we do. And it's incredibly exciting to listen to, the perspective from the patient and the NIH bioresource in terms of the data. But we're really excited because we have been focusing on trying to identify medications to treat aspects of inflammatory bowel disease, and the kind of data that is now being made available is really essential to the work that we're doing. And so today I'm going to talk a little bit about exactly how we use the genetic data and why this matters. So if we can go to the next slide, you can just see our disclosures. And then let's move to the slide after that. So we wanted Victoria to kick off because she shared with us that the patient perspective is incredibly important to her because of her nephew. Her nephew was diagnosed at age 12, and they gave him several different types of treatments, and none of them really worked. And that was, of course, very challenging. He was extremely ill. He didn't respond to these treatments. He was terrified of having the biological treatments because of the IV delivery. But it turned out that Remicade worked and continues to work for him, and he feels well and is able to live a normal life. But there are, we know that there are many children and adults who continue to wait for medicines that will meet their need, and it hasn't, they aren't there yet. And so, certainly what we're trying to do is to really think about, what is the best way to treat that. If you can move to the next slide. So as you know, Crohn's disease and ulcerative colitis are both relapsing, remitting chronic inflammatory diseases of the GI tract. And we call that inflammatory bowel disease. They can involve, I'm probably preaching to the choir here, and they can involve any segment of the GI tract, but most commonly the terminal ileum and other colon. And the inflammation is typically segmental and transmural. The inflammation starts in the rectum and can extend to variable degrees to proximal segments of the colon. It's continuous and limited to the epithelium. But it also may be accompanied by extra-intestinal manifestations, which include inflammation of the joints, eyes, skin, liver, and biliary tree. Globally, the prevalence of IBD is about 0.75%, which means, like in a company at GSK, we'd expect 750 employees to suffer from IBD. Next slide. Sorry, just managing my thing here. A condition like IBD takes, developing a medicine for a condition like IBD takes a long time and is incredibly expensive. And not only that, many of the potential drugs never make it to the market. And it turns out that the drag of this attrition really brings our productivity down, making the R&D spend for every drug that is approved over $2 billion. And you have to ask ourselves, why do our efforts as research scientists fail so much more often than they succeed? And the reason is because we don't really understand the diseases very well. Pharma has been focusing on creating drugs for unmet needs and diseases which are really, really difficult to treat. And even for IBD is that we do, we have had some effective medicines, but it doesn't work for everyone. And so it's important to really try to identify why that is. Pharma's been, for decades, we've been doing kind of all we can to squeeze the new insights from old drugs, the existing mechanisms, animal model. But it turns out that the biology can be really complex. And so we kind of been feeling our way until in darkness. And I think what we're finding is that genetics is giving us a really fresh insight into disease that will enable us to really revolutionize drug discovery. And I'm going to talk a little bit about how we do that. If we go to the next slide, I'm going to give a little bit of information. I'm just going to give a little bit of background so that as I go into my talk, you'll understand a number of the items. So I'll talk about phenotypes. These are kind of that's information about a person's health. Genotypes are going to be the genetic information. Samples, those can include tissue biopsy, blood, stools, urine, saliva. Laboratory measures, which might be things like cell counts or biochemical measures like cholesterol, CRP, blood pressure. And then genomics will be kind of more additional measures of including transcriptomics, proteomics, and metabolomics. If you go to the next slide. So the key to the revolution that I was talking about earlier is that we have found that using genetic evidence to help us make better choices about where we start our research is a really important aspect. And, drug discovery usually starts by finding a key piece of disease biology that we can manipulate to explore whether revving it up or shutting it down would help a patient. And we call that a drug target. That drug target is usually a protein produced by our genes. And so our first challenge in the drug discovery odyssey is to figure out which gene amongst the thousands of possibilities that we're going to work on. And to give an analogy, around the time that Google was conceived, the scientific world got really excited because we thought that we could, we thought that the Human Genome Project was really going to, help us find the drugs. And the hope and the hype was that this, the sequence of our 3 billion base pairs would deliver the blueprint and kind of identify, okay, these are the diseases and the drugs. And it turned out that it didn't work like that. Initially, we had a lot of information, but no reliable or fast way to understand it. And so now we've had amazing new technologies that are really giving us fresh and valuable insight. So for instance, about a decade ago, we only knew a handful of genes that were involved in common disease, but now we can literally, now there are like 50,000 of those genes, and that is giving us really an amazing new place to start. So it turns out that knowing if there are differences or variation in genes affects our risk of disease is a very valuable way to identify which proteins make good drug targets. And we demonstrated that drug targets with human genetic, I'm sorry, you can go to the next slide. We demonstrated that drug targets with human genetic evidence are actually twice as likely to result in a successful medicine than those without the genetic evidence. You can see in the slide here the along the x-axis, you have, the genetic targets, and you can see that as you go further and further along, those that have genetic evidence go farther into drug development. So this is just an incredible new tool for us, and this has really impacted the entire industry, changing the way that these drug targets are discovered and prioritized. So within GSK, our drug discovery efforts focus on identifying these genes that or drug targets that have more genetic evidence. We're hoping that by increasing the proportion of early stage drug targets with human genetic evidence, that we can reduce the costs for each successful drug. And that's really why the genetic data is so important. If you go to the next slide. Just a click that shows, kind of where many of the failures. Most of the time, when we have a failure, it's because the drug doesn't work, and that's kind of why we need to understand better of the biology. Most of it is that you can see there in the orange that most of the drugs that fail are because they aren't effective. That they don't do anything. If you go to the next slide. So here we talk about a gene. It's made up of exons that code for the portions that make proteins. If you click again, at the lowest level, our DNA is made up of these, and again, 3 billion base pairs called nucleotides. Many of these have small differences, which we call single nucleotide polymorphisms, or SNPs. Most of them don't have any effect, but some of them change the way that proteins are made, that can give rise to differences in how tall we grow, the color of our hair, and how likely we are to develop osteoarthritis or IBD. And we can measure these genetic differences. If you go to the next slide, next click, one more. In this example, we're testing, and click one more time. We're testing whether the frequency of the variant, the single nucleotide, the SNP, the single nucleotide polymorphism, is different between people that have a particular disease versus those that don't. In this case, we're looking at osteoarthritis. In this case, there's a pretty big difference that people that have osteoarthritis have an increased frequency of the C allele. And that's a big difference. And we conclude that there's something about this region of our DNA that's influencing our risk of osteoarthritis. We don't usually know in advance what genes or parts of the genome that are involved in disease. So we have various ways to quickly and cheaply measure the genetic differences across the genome and test each of them. And that's the kind of data that's available at the NIH bioresource. If you go to the next slide. So some of the opportunities that we think are really afforded by the patient level data and the gut reaction HDR hub helps us identify drug targets. What we mean by target identification is that we're using the genetic data from the large populations of patients like are available here to identify the genes that may be associated with IBD, ulcerative colitis, or Crohn's disease. And that gives us a clue about the genes that could be most effective as drug targets. The second thing we might do is to, once maybe we found a region of the genome, we might try to understand the genomics data that's generated in this hub to understand, including things like the affected disease tissues, to understand which cells express which genes, which cells are and which genes are altered compared to healthy, and how a particular variant might affect an individual's expression. And we utilize data from here and other publicly available sources. If you go to the next slide, some of the other types of things that would be done with this type of data would be is kind of stratification and focusing on the unmet need. As we've already shared, there are already medications for IBD, ulcerative colitis, and Crohn's disease, and so it's important for us to kind of pick up where those leave off. And by understanding deep clinical data, we can identify subpopulations of patients with a high unmet need, or explore what might be the cause, comparing those populations to those with milder disease or healthy controls. We might want to understand why people don't respond to the standard of care, or they might have more rapidly progressing disease, or they might have more severe disease. And this subpopulation information is actually really not very common, but really critical for us to take a particular gene that we might say is associated with a risk of disease and understand, what particular aspects of the disease is this gene going to have the most influence on. Furthermore, I've been helping to develop a capability called recalling folks to either collect additional tissues or to do additional assessments. And these are really, this kind of recall studies are really, we feel like are going to be an incredible innovation for us to really understand at a very deep level why some people have more severe disease and others do not. And so usually we start with a very targeted hypothesis that helps us, that the data that we have so far is suggested, and we use that to really understand why we take that hypothesis, want to be able to engage with patients, obviously with their consent, and to learn more about that particular hypothesis that we feel is critical to developing the medication. So in summary, I just want to say that all of this is really, I hope that this is giving you a sense of how this data is making such a difference in our ability to start with a particular gene, and which I've explained is, we feel like, is one of the biggest new innovations in drug discovery, and to use that gene to get an insight into the biology and then really use that to try to develop the next generation of medicines. I'll just stop there. Thanks.
So thank you, Meg, and also thanks to Victoria, who I know has put a lot of effort into preparing the presentation, even if she wasn't able to contribute. And obviously you see here the others who also have. Do people have questions for Meg? Clearly, our interaction with industry is absolutely vital for the sustainability of the hub, and indeed also for developing the new therapies that people with inflammatory bowel disease need. But absolutely essential that we carry all of those involved in the hub with us as we move forward with the collaboration with industry. So one of the questions here that's just come up, is there any difference in the way industry applies to use data? I may pass over to Neil in just a second, but there is no difference in the way that the application process for industry, but we perhaps will be more inclined to push that through to the National Steering Committee for review. Neil, I don't know whether you want to add anything there.
No, it is very, very important that we don't do anything weird or unusual with our industry partners. They get vetted by the same patient advisory committee as everybody else. As Tudor particularly said in his response to Sarah, it's very important that we take people with us. And I asked a different patient advisory committee yesterday, did they ever refuse? And they said yes, they did. Where the steering committee or whatever said we'd like to support this study because it's going to be really good, and they said no, this is just horrible, we're not doing it. But they also said on the flip side that they had actually proposed accepting something that the steering committee were rejecting, because they say, we are the patients, we need these drugs, we know who makes them, it's not you. It is the industry who make them and not you academics. So it was quite interesting that they, although they were generally in consensus, they were prepared to take the lead both in rejecting and proposing projects. But it does go through the same panel, the same 5 safes idea. So we know who they are, we know what the project is, we know where the data is, we know what the institution is, all that stuff is the same.
Thanks. And I should say we're learning all the time. And again, I think our collaboration with GSK and other partners has been very important in establishing how to take this forward, and involving patients and public and other interested parties as we go through the data application process. Meg, I don't know whether you want to add anything from your perspective on how easy it is or the challenges of applying for data.
No, I felt like it was fairly easy, and I think the key thing was just, I think once we explained kind of how we wanted to use the data, the questions we wanted to ask, I think it was a fairly simple application.
Thank you.
I did see there was another question in there, another question in the Q&A. Should I attempt to answer that?
Yes, is this from Alex just come up, or was that another one?
Sorry, and I had to plug my phone in before it started to die, so I'm kind of holding it in a very awkward way. So, okay, wait a minute, let's see, maybe I missed it. There was a question that asked if there were any, I think I saw a question that asked if there were any particular types of data that were more valuable.
Yeah, it's just come up. What data types are more important than the others?
Yeah, I think, should I,
yes,
do you mind?
Yeah.
Okay, so no problem. So what I would say is actually the most important, I would say that we, through efforts like the UK Biobank and some of the consortium and publicly available efforts, we've been able to access information that basically says, does a patient have disease or not have disease? But what we've really struggled to get access to is really detailed information about a person's course of disease, the medications they've taken, the various procedures they may have had, even things about their function. So what I would say is that type of data is incredibly helpful, and unfortunately, what's oftentimes we can get access to small data sets, but in order to get access to data sets that are large enough that we can really ask a question, does this particular gene associate with a more severe form, or people that don't respond to a certain medication? So that particular type of information is super important.
Yeah, and I think there was a question earlier about what is linked data, and I think it really is the linkage between the genetics and the clinical data that really is powerful, and what we're trying to achieve through gut reaction. So a comment or question about there should be more advertising regarding data sharing. Many people, especially people who are not used to modern technology, are worried what is done with their data. Those patients are all so important to groups and research. And I think this does come back to some of the early discussions really around communications, about the power of the data that we're collecting and making accessible, and the processes that we have put in place to ensure it is secure and used appropriately. And then there was a question from Alex, what can academics do better to accelerate the genetic association through to drug target, through to drug pipeline?
So probably one of the biggest aspects is when papers are published, the summary results are oftentimes attempted to make publicly available, but many times they're really not in a format that can be easily used. And I would say that is actually one of the biggest challenges. What we do is really a big data problem, and that is that we're really trying to leverage any internal data that we create, data like the NIH bioresource, and data that's published to understand everything we can about IBD. And when the information, the summary data is published, if it's not sort of put in a public repository where it's really well curated, well harmonized, that can really hinder the ability to leverage that data for drug research.
And then one final question. Is the data used for AI-based applications? And I can answer that. Megan can come in. Absolutely. The data at this scale, then we absolutely want people to be able to use it for machine learning and AI-based applications. I think that's something shared by industry, Meg.
Yeah. Yes. Actually, we have a researcher as part of our application that is hoping that we'll be able to use some artificial intelligence, machine learning methods to understand kind of patient subgroups. And that would be patients that look similar to each other, either in their presentation or the type of medications they take, or the severity of procedures. So yes, that is definitely done as part of this.
And then finally, and a question which I think illustrates how informative this session's been, a really good question. Should patient summaries on published articles be encouraged so they can be circulated by a patient community? And I think we're very good at lay summaries at the sort of application and project stage, but we don't tend to do that around the publication. So I think that's something that we should take away and think through how we might at least do that through gut reaction. So I'm going to draw things to a close now. I hope that people who have attended now have a better understanding of gut reaction, what we're trying to achieve, the breadth of its activities, and how we will make it sustainable. I want to finish by thanking all of the speakers for some really excellent presentations, but most of all for thanking the participants for the questions that they've been putting to us. These are really important to highlight the issues that are important across the wide-ranging audience, and indeed to inform how we will develop gut reaction in the future to make it usable, to make sure that we deliver our objectives, and to make it sustainable. So on that point, thank you to everybody who has attended. I wish you all a good weekend, and thanks again for your participation.